Thank you very much. Very glad to be to participate in this session. And this particular session has been specifically carved out to reflect what what innovation technologies that have been spearheaded by innovations done by entrepreneurs, startups, and the early medium scale industry that can contribute towards the challenge of delivery of the vaccines at the last mile. See, when you look at in the beginning, we had challenge to prevent COVID spread, and we were looking at sanitation, PPE kits, masks, etc. We also went through a phase where we were wanting to detect COVID patients and likelihood of infections. So we were working and we were looking at solutions which are diagnostic kits, imported diagnostic kits, indigenized diagnostic kits, and availability of such diagnostic kits, testing centers, and covering as much as population of India. Then we also started covering the therapeutic arm. There was a phase when we were looking at repurposing of existing molecules and drugs to making those drugs, making available not only for India, for the entire world, and the vaccine initiative. So we have, in the last 10 months, we have reached to a position where we have been able to move from as a front runner, as a contributor in many of these sections. And some sections where we were initially started as net importers, we have become self-sufficient, Atmanirbhar, as well as we are importing, including PPE kits, the drugs, hydroxychloroquine, remdesivir, and N95 masks. This phase, now we have moved into where we are looking at vaccine delivery to the last mile. And the very fact that we are today talking about and having this conversation is because there are expectations. Expectations not only from Indian population, but world at large. Looking at India, can India contribute, and innovations can emerge from the Indian ecosystem, which can help in the delivery of the last mile of the vaccines, be it candidates which are emerging from India or outside India. So today I'm very happy and pleased to introduce, we have a interesting panel in which there are few technologies which have been, which are on the anvil of development, while there are a couple of them which are already developed. Yeah, so we have like professors who have contributed and created knowledge in the academic setting. We have young innovators, startups, medium scale, which have actually delivered and created the technologies, as well as both from India as well as abroad. So in the next 1 hour, what I'll do is I'll provide each one a 10-minute exposure where you can share, if you have slide deck, to the forum, and I will introduce, and then maybe we can begin with that. May I introduce Dr. Atul Sardana first, and he can show. Atul is a surgeon, and he has been associated in the innovation and delivery of solutions, including laparoscopy, trocar, coated hernia mesh. But today he will be specifically talking about the safety syringe that he and his team under Alpha Corpuscles have developed. Over to you, Atul, and you will have 8 to 10 minutes to share your invention with us. Thank you.
Thank you, sir. Okay. So what if I was to say that, there is a technology available which can deliver the COVID vaccine to the last mile very safely, very effectively, without generating any hazardous biomedical waste at a much lower cost, and can deliver twice as much vaccine uh, per individual provider than what we are currently planning. So as we prepare to undertake one of the world's largest vaccination drive, a lot needs to be considered in selecting the most appropriate vaccine delivery technology. India needs to cover a population of 1.3 billion, and currently media reports indicate that two shots would be required. And the government is allocating a budget of estimate around $7 billion. We are estimating that two vaccines, $2 per vaccine would be required, and another $2 in the infrastructure cost. So we are putting a total of $6 per individual over here. Now, we need to move up the value chain for COVID-19 vaccine delivery. What we are currently looking, as I heard the regulator talking about safety syringes, I'm very happy that the regulators are aware of such technologies. But unfortunately, current focus is on AD syringes, which is a very old generation technology which just locks the syringe, or you can break the plunger after use, and it does not address to the needle. And it is actually the needle which carries the infected blood from one patient, and it is the reuse of needles which is the cause of infections rather than the syringes itself. Obviously, we have other technologies such as pre-filled syringes available, but then there are certain problems associated with them. So I am here to present to you a technology which is 10 steps ahead for quick, safe, accurate, and cost-effective COVID vaccine delivery. Now we're talking about a very special safety syringe which has been developed only for vaccine delivery. It is a cartridge load safety syringe. Here, the same cold chain space can carry 10 times more doses as compared to a pre-filled syringe, thereby reducing the cold chain cost per dose to 10%. Also, it reduces the associated failures by packaging in smaller boxes, which are easy to carry to remote areas. Because we are looking at delivering a vaccine to the farthest areas in India in a very short period of time to get maximum coverage. So if we estimate the cold chain cost per dose to be 60 cents, it would be reduced to 6 cents rather than 60 cents. This technology saves 10% of the vaccine cost by reducing the need to overpack and compensate for the filling and injection losses. As Dr. Mahapatra was speaking in the last panel, he said we haven't considered, breakages, filling losses, and so this is a technology which could save those losses. He had estimated 20%, but I am putting it at a conservative 10%. So we save the estimated cost of vaccine per person is $4. We can reduce it to $3.6 immediately. It also saves on the breakage, which is estimated to be around 1%, because the cartridge over here is a polymer cartridge and not a glass cartridge, though we can develop the same technology in glass if there are any regulatory issues. But the polymer itself has been tested to minus 50 degrees and can further be tested to minus 70 degrees in case we are looking at an mRNA vaccine. It would save 20% on the freight cost due to lower weight of the cartridge and as compared to the vials and the pre-filled syringes. It would take 50% less time per injection as compared to vials and syringes. Hence, more number of people can be vaccinated in the same time. So same staff, same hospital can inject twice as many patients per day, thereby reducing the coverage time to half number of days. There are zero chances of contamination while filling, so that there is no residual infection to take care of after the vaccination drive. A lot of vaccination drives in the past have actually witnessed, reports coming up saying there was a vaccination camp in so-and-so village, and 1 year down the line, we find the entire village is HIV positive. So there are no chances of this thing happening. It also ensures 100% consistency in administering the dose as compared to syringes, thereby reducing the variability in the immune response, which could be due to the dose variability. It has a 100% safety integrated into it, which prevents reuse and accidental needle stick injury. Now we all know that reuse of syringes and accidental needle stick injury puts a direct cost of more than $600 billion worldwide each year to manage the diseases spread by bloodborne infections from such unsafe injections such as HIV, hepatitis B, and hepatitis C. India itself has about 78 lakh infections every year from unsafe injections, 3 lakh deaths, and a direct medical cost of 700 crore rupees. If you look at the global figures, is 1.2 million early deaths. That, if you put on a time scale, is one death in every 24 seconds, which is higher than the toll of most of the lethal diseases that we have. The use of this syringe requires minimal or no training. It does not require any user participation to activate the safety features. Once the dose is complete, the safety features are deploy themselves automatically. There is no requirement for sharp disposal boxes. Now imagine the number of people we need to vaccinate. We need to carry the empty sharp disposal boxes, carry them back with us. That is going to save time, because once the syringe locks up, there is no part or no sharp part which is there which could injure anybody. So you could simply carry the biomedical waste in bags and then have them disposed as per the guidelines. So there is an effective reduction in the biomedical waste because the boxes go away. The design is 100% tamper evident, so you get unparalleled safety and security. The design is compliant to WHO and the relevant ISO standards, and the cartridge itself can go into existing filling line for all pharma manufacturers. The cost of this is estimated at less than 15 cents. This technology is now ready, available, and it has been developed with financial assistance from BIRAC, and we have done a clinical trial covering 2,000 patients in India with assistance from ICMR. For any further details, you can contact us. I would also like to show you the syringe itself. This is the cartridge. This is the cartridge. This is the only part that stays in the cold chain, not the entire prefilled syringe. And it takes almost 1/10 the space as compared to prefilled syringes, and it provides the safety and efficacy of a prefilled syringe or a prefilled safety syringe. It's a single step to load the cartridge inside. These cartridges can go into existing filling lines, which are there available with the pharma manufacturers. Thank you. I'm open for any questions now.
Thank you so much, Atul. I think you clearly brought out that a technology intervention like this can potentially save the precious doses of vaccine by reducing as much as 10% overages, which are required for distribution in the unit packages, as well as the safety syringe can potentially prevent the inadvertent needle-related injuries when we are looking at mass vaccination at
Yes.
such a large volume. Thank you for bringing it up.
And 1/10 of the cold chain space, sir, which of
Absolutely.
course is one of the biggest pain points currently we have with this vaccine.
So that makes me bring and introduce our next speaker, Dr. Samit from Plus Technologies. He and his team has brought in and working on the energy excess, and they have been able to build a first-in-class Mira cradle for babies that has been able to keep the baby cool in up to three days. And they've also created a product for fine chemical deliveries in the pharma sector, the Selfshare, and maybe he will want to showcase and reflect more how it can facilitate the last mile delivery of the vaccine. Over to you, Dr. Samit.
Thank you, Dr. Manish Devan, and thanks BSMA for this opportunity. Right. So what we as a company have been specializing in is temperature control across sectors. So as Dr. Manish mentioned, we did a product for keeping babies cool, that is treating babies born with birth asphyxia. So we have done medical devices, we are looking at logistics, buildings, etc., wherever we need temperature control. And we do this using special materials that we excel in, which are called as phase change materials. We have looked at neonatal death, we have looked at building energy efficiency, we have looked at food wastage. All of it needs temperature control, and current focus is on the vaccine wastage or the vaccine delivery, which could need different temperatures, right from -70, as we are talking of the Pfizer vaccine, for example, or a lot of other vaccines at -20 to -30, and the traditional vaccines, which we have always been used to and delivering and administering at the 2 to 8 degrees Celsius range. I will not spend time on the challenges. We've talked a lot about it through the day. What we are now looking at is looking at freeze-safe vaccine carriers for a lot of the vaccines which need to be at 2 to 8 degrees. And when you use conventional ice or ice packs, the chances that the vaccine goes below 2 is very high, and that itself is effective. So we have used our materials to create a box which will always keep the vaccine above 2 degrees Celsius. We are now looking at -20, and we are also looking at -70. And these are possible using these materials, which we define as phase change materials. They change phase from solid to liquid and liquid to solid. Water is the best example that we are familiar with. You freeze water, you get ice. You melt the ice, the product that the ice is put into becomes cool because the heat is either absorbed or released. But that happens at 0 degrees Celsius. So we have designed materials right from -30 currently to +100. And depending on what temperature we want to maintain, we use the appropriate phase change material. And these are also often referred to as thermal batteries because they store thermal energy. It's essentially a thermal battery, a temperature regulator, and a good heat exchanger, the way you design the whole thing, and that together forms the phase change material. So the three most common temperature ranges one is looking at is 2 to 8 degrees Celsius, 15 to 25 for a lot of pharmaceutical products, -15 to -25, and now, as I said, -70. And for these three different categories, we use three different phase change materials as against either cold water, dry ice, or gel packs or ice packs that people typically have been using. So we have developed several solutions. You can see some pictures on the right. The bag that you see is currently being used by the likes of, let's say, Dr. Lal Path Labs and others to start collecting the samples that you're doing for testing. It has a backup of 10 to 12 hours and can maintain a temperature between 2 to 8 degrees Celsius. We are now working on a vaccine carrier, which we hope to launch in 2 or 3 months' time, which will ensure that the last mile vaccine delivery will never be below 2 degrees Celsius, especially when you look at polio vaccines, you look at DPT, measles, etc. They all need the 2 to 8 temperature. We are looking at hard case boxes, which can be for as long as 24 to 48 hours, and even beyond that, we have tested till about 120 hours. This product is under the brand name Selfshare, and in the last 2 years, we have started working with a lot of pharma companies for the first mile, where from the manufacturing unit till the distributor, the vaccines are packed and then distributed at the right temperature. So for example, if you're shipping from, let's say, Hyderabad to Africa or Hyderabad to South America, you're looking at 4 or 5 days of temperature maintenance in a passive way. These have been tested as per the standard test methods followed with backup durations of up to 120 hours. These boxes have also been integrated with a lot of smart sensing to real-time monitor the temperature, the location, the vibrations. The box shouldn't break, the vaccine vial should break. Very often light penetration could also affect the medicine that is inside, for example, or the vaccine. So these can easily be monitored and notifications sent on an app on your phone. These are some validations I'll show you to indicate the kind of temperatures these have been tested against. The red color tells you the atmospheric temperature varying from 20 degrees to 35, and you get a backup of 120 hours, and the temperature never goes below 2 or above 8. This is at a -5 degree ambient. The red color again is showing you a temperature which is there, and the blue color, which is the temperature that is maintained inside the box. Again, a backup of more than 115 hours or so. This is a customer validation that was done with varying temperatures, right from +20 degrees to as high as +40. But the temperature inside the box remains above 2 degrees Celsius and below 8 degrees Celsius for as long as 150 hours. At the -15 to -25 range, in an ambient of 35 degrees Celsius, for example, the temperature is still between -15 to -20 for about 120, 130 hours. What we are looking at now are better insulation materials. We are all familiar with the vacuum flasks that we use. So we are looking at using vacuum insulation panels to be able to create the box, which could be lighter, which would be probably much more easier to handle, less space. And as you see in the picture, something like, a polyurethane foam that we are used to, 145 foam can be replaced with a 35 millimeter vacuum insulation panel. Costs, of course, will be a challenge, but then a lot of vaccines which are very expensive, for example, or medicines which are expensive, it would really prove useful. We are looking at more user-friendly ergonomic designs for the last mile delivery, how it can be, for example, mounted on a two-wheeler more easily with different chambers that can be done. So a lot of ergonomic designs are necessary to ensure that the work becomes very easy for the last mile worker that we have, who has to go from door to door and administer the vaccines. This is another last mile delivery solution that is being done, extremely rugged and using phase change material liners. The yellow green color that you see is going to be a liner inside the box to ensure, along with the conventional ice packs, which is in the blue color, to ensure that your vaccine remains above 2 degrees Celsius at all points. So this is being done in partnership with Path, and we hope to be able to launch it. And most of the COVID-19 vaccines also, while it will be stored at -70 or -20, the last 24 hours will always be at 2 to 8, because you cannot quite inject something at -70 degrees straight into the body, right? So there would be a huge requirement we foresee for the 2 to 8 degree solutions at the last mile. What we also did for the Tata Medical and Diagnostics, for the CRISPR kit that they have launched, the Tata MD Check Feluda based kit. So this is the box from the outside, looks like any other box, but inside we are able to maintain the -20 degrees Celsius using the phase change material. And one section has a 2 to 8 degree for the reagents that they carry. So this has been done in a record time of about 2 months, working very closely with Tata Medical for getting the solution. In fact, Delhi today said that they will start using the Feluda kits effective today or tomorrow. So we see that it is really possible to use advanced materials, move away from dry ice or move away from regular ice packs to ensure the right temperature maintenance. We've been working with DBT funding for a lot of our medical device products. We've been working with UNIDO as well as CII on a lot of these projects, and I'm open to any questions.
Thank you, Dr. Samit. Would you also like to let us know about the price point of this particular delivery device?
So we are looking at prices when you're looking at, let's say, something like a 5-day or a 6-day maintenance, and you're looking at a capacity, vaccine carrying capacity of, let's say, about 7 to 10 liters. you're looking at a price of 3 to 5,000 rupees for a box. So when you're shipping vaccines from Hyderabad to, let's say, South America, as I mentioned, and you want a 5-day duration, that's the kind of cost you may be looking at. When you look at the reusable solutions we are talking about for the last mile, we are again looking at something like 1,500 to 2,000 rupees, but then it's reusable, so it's going to be pretty economical in the long run. So costs will vary between 2,000 to 10,000 depending on the size and the durations that you typically look at.
All right. Thank you so much. I think connected with this, in a similar thought process, I would now introduce Mayur Shetty, who is the founder and CEO for Black Frog Technologies. His team is also, his startup has also come up with a solution which can carry vaccines and perishable biologics in the ambient temperature. Over to you, Mayur. Yes.
Yeah, thank you. Hi, hello everybody. I'm Mayur Shetty. I'm CEO of Black Frog Technologies. We are a technology startup company based out of a small town called Manipal in coastal Karnataka. A little bit of background on our work. In about 2017, BIRAC funded us to develop a portable medical grade system to address the last mile problems associated with the delivery of vaccines. And I'm pleased to state that we're now live in 3 states in India, and the product has won us about 6 national awards, and we've represented India in 2 international events as well. I'll just briefly talk about the context of the problem that we're solving. It was quite extensively discussed today. The vaccine supply chain is very well understood, but BIRAC in 2017 entrusted us with finding a viable replacement for the ice boxes or passively cooled containers, which have typically and been used over a century now for transport of all thermally sensitive biologicals. The problem is that while temperatures drop below 2 degrees Celsius, there's also no accountability or form of active monitoring inside this particular chambers. India has had a very successful universal immunization program. It's deemed as one of the most successful public health interventions all across the world. But there's also a slightly darker side to this, where when we go about administering polio vaccines, we know that the recommended dosage is about 3 times, but there have been reports of going 8, 9, 10 times as well, because we know that the vaccines that we've been administering could possibly be thermally degraded before the time it's administered. The Immunization Technical Support Unit under the Health Ministry claims close to 25% of all vaccines go to waste before they're administered, and almost 73% of this degradation happens in the last mile, and that's predominantly because of use of ice boxes. So while we've had the factors of time and resources in place for mitigating any other vaccine preventable diseases in the past, that's not the case right now with the COVID-19 vaccine in the horizon. It's not only a limited resource, but to give an unviable vaccine while people are expecting a magic bullet is probably the nightmare scenario. And that's what we're trying to address with a actively cooled portable device. And I'm very proud to announce that we've been successful at it over 3 years of R&D on this, where we've developed a portable and rugged battery-powered system that can maintain a preset temperature between 2 and 8 degrees Celsius for up to 12 hours in the field. The device weighs just 5.5 kilograms. It fits into a backpack, can be worn as a sling bag as well. We've designed the cold chamber in the same form that a typical blue box commissioned from UNICEF is able to carry. It's about 2 liters in capacity. That's usually the day-long transport limit. And 2 to 8 degrees Celsius is what we focused on because, just from the earlier conversation as well, Dr. Harish Iyer mentioned about Pfizer's vaccine and Moderna's vaccine as well. All of them, regardless of what the storage temperature is, the last mile is still going to be 2 to 8 degrees Celsius. Pfizer's vaccine is thermostable at 2 to 8 degrees for up to 5 days. I believe Moderna's is up to 30 days as well. So end of the day, it's going to come down to passively cooled devices or ice boxes that have to transport from primary and sub-health centers till the point of administering. And the reason that we worked on a solid state cooling, actively cooled product is that it brings about two distinct advantages. One is the fact that there is uniform cooling all throughout the cold chamber, which means there are no hot spots or cold spots. All of the vaccines are assured a temperature between 2 and 8 degrees Celsius. And secondly, and most importantly, a ground reality when you administer vaccine is that an ANM or a healthcare worker has to open the device every now and then to retrieve the vaccine to administer, which means every time you open the vaccine, you subject all of the contents to the ambient air. So this enormous amount of freeze thaw cycles happening. So now that's where an active cooling device comes in, where every time you shut it, our system is able to bring the temperature, stabilize it back to safe limits, that's 2 to 8 degrees Celsius, over 96% faster than a passively cooled system can. And this is very critical because uh, apart from thermal degradation directly because of prolonged periods of exposure, it's freeze thaw cycles which degrade the biologicals as well. And we've had some successful results from our field trials that we've been implementing over the past 2 years. Just like to point out one of them where we're working with Kasturba Hospital for transport of routine vaccines. So we use these data loggers in their system, and we carried out the same trial with our device as well, where wireless temperature loggers were used in the system. And then the blue line is the 2 degrees Celsius mark, and the red line is the 8 degrees Celsius mark. So on the left, you can see that the ice box commissioned from UNICEF is constantly dropping the temperature below 0 degrees Celsius and freezing the vaccines. So this translates to about $7.5 million worth of vaccine wastage every single year in India alone because of using ice-based system. And this is a pre-COVID scenario. So we can totally understand the kind of ramification it will have when we'll have a temperature control dependent COVID vaccine in the horizon now. On the right, you can see our system is able to maintain a very stable 4 degrees Celsius platform with an error of less than 0.5 degrees Celsius. And we developed this about mid 2019, and we started deployment towards the end of the year. And I'd just like to pinpoint some of the use cases. So our earliest customers were Selco Foundation, and one of the deployments was a very interesting use case in a remote hospital called Sargur in Mysore, where they're using our device to transport routine vaccines to a local tribal community who are otherwise inaccessible and kind of cut off from civilization. So now they're able to transport vaccines and immunize twice as many kids every single day because, due to change of vaccine transport from ice boxes to an actively cooled device. They have a mobile health unit with a solar powered system on board. So they're able to reuse the system every day, and not to mention they're able to bring back the vaccines that are left over at the end of the day. So they're claiming a savings of about 13,000 rupees worth of vaccines every single month from use of our device. And towards the beginning of this year, when we were scaling up our production capability, that's when the COVID-19 outbreak hit us. Fortunately, we were at the right place at the right time because COVID-19 specimens, the nasal and throat swabs require the same temperature. And we gave out a couple of the units to the district health administration as well. So they found the product very utilitarian in that there were a lot of concerns about false negatives arising because of samples degrading while transport. So that's how we kind of picked up traction. And now we're at a point where we're delivering to Jharkhand and Orissa for transport of veterinary vaccines as well. Our objective right now moving ahead is to scale up our production capabilities so that we are able to cater to the vaccine requirements that is upcoming over the next 2 to 3 years right now. Our focus would remain integration into the electronic vaccine intelligence network, EVIN program, which has been very successful by the UNDP and MOHFW. So this provides clear traceability and temperature RTM for up to the cold chain point or the primary health center. So now we have a battery powered system that is integrated to a dashboard. So there's a lot of potential to integrate our API with the EVIN network itself. So this will provide end-to-end traceability for MOHFW to ensure where the vaccines are coming from and exactly where it's going. The location and other vital statistics that we're transmitting will help with it will help with figuring out the immunization coverage, where the device is going, kind of understanding and helping with the mapping the outbreak of disease over time as well. What areas are being immunized, what are being left out. So all of these are very crucial and vital data for informing value chain decisions. So this is part of our plan right now, and BIRAC I'm very proud to say that they've just funded us with an ongoing project right now where we are upping the R&D on the product. So this is a battery-powered system that is going to the point of administering, which means there's a lot of potential for incorporating a lot of existing technology on board. For example, NFC and RFID scanners. So US FDA and CDC had thought about integrating 2D barcodes on each of the individual vials. So this is a program that is just picking up force right now. And our vision right now is to have a vaccine schedule card, which is already being implemented with data on what vaccines you are due and what vaccines you have already been administered. And then there is vaccine vial information on each of these particular vials. So every time you administer a vaccine, you touch the vial to the device, you touch the patient card to the device. This card can be a NFC-based card, which is the current practice, but I think MOHFW is moving towards making the Aadhaar program itself compatible with this, so that on a cloud level, there is clear hand-holding of who got which vaccine from which manufacturer, which lot. So this will provide a lot of value when there is an adverse drug reaction arising from a particular vaccine. It will help the pharmacovigilance team in understanding where the drugs are going. And it will ensure that there are no counterfeit drugs or any black market arising, which is a real possibility with COVID-19 vaccine being a limited resource now. There's no risk of hepatitis A or hepatitis instead of hepatitis B being administered by mistake, because the device itself is going to be providing alert mechanism. Yeah. So this is, I'm going to just abruptly stop at this point. We're just, as part of our longer vision, we're hoping to be a part of the bigger program of delivering efficacious vaccines to the farthest corners of the country. Thank you, Dr. Devan. Thank you for providing me with this opportunity. I'd love to take any questions. Thank you, sir.
Thank you, Mayur. I think that was a very clear presentation. And I think both Selfshare and Emvolio presents a view that precious vaccine wastage, which would be like shameful to have that in case of COVID vaccine, where we need to cover such a large population, and we are looking at more than one doses to cover the population. And solutions like this could actually preserve the wastage of the vaccines. Even the pre-filled cartridges, which Alpha Corpuscles Atul mentioned, I think these solutions create value and create opportunities which can implement and bring value for this. I now move to other interesting case, and I'm very happy to introduce Pavanshu, Dr. Pavanshu Mohanty and Laura, where they are providing a solution with excipients that can actually obviate the need of carrying vaccines at low temperatures. And it's very exciting, exciting solutions by Akara. Is that the right way to pronounce it, or you want me to pronounce it as Akara Technologies? So over to you, Dr. Pavanshu and Dr. Laura.
No, Akara is the right word, but
right.
it has been anglicized. But Akara is the correct word as Indian origin. You can understand that.
Kindly go ahead. Stage is yours.
All right.
Well, good afternoon, everybody. So I'm Dr. Laura Bronzer, and I'm the program manager for Akara. And I'd like to introduce Dr. Pavanshu Mohanty. He's the founder of our company. So Akara, as was stated before, is interested in biopreservation as a technology, particularly for the global market. So what has been discussed quite heavily today is that cold chain logistics are going to be a major issue in the distribution of vaccines, in particular COVID. In the case of a pandemic where you want to be able to reach every community, including the rural communities, getting vaccines to the area where they've been maintained at the proper temperature is crucial. This is going to be extremely evident with COVID, where a large majority of the candidates require storage at very low temperature extremes, so -70, -80. And those type of extremes, currently, there isn't the infrastructure to support them. There's also financial limitations when it comes to cold chain distribution of vaccines. So when you consider the pharmaceutical industry as a whole, it cost about 18.6 billion in 2022. That's an estimate for the distribution of vaccines that require cold chain. And there's thought to be about 35 billion lost in revenue due to temperature excursions and the impacts those have on clinical trials and the loss of product. And finally, because of cold chain requirements, transportation has to be done by air transport versus sea transport, and that costs about 700% more. And what this results in is that vaccines cost more than what they could cost, and that reduces the ability to vaccinate large communities or rural communities. So there was a study conducted where they modeled just the removal of the cold chain from the last mile, as you've been discussing. What would be the cost to vaccinate an individual if you didn't have to have cold chain during that last mile? And that actually reduced the cost of vaccination from 24 cents a person down to 12 cents a person. So just that last mile had the impact of reducing the cost by 50%. So where our technology comes in is our anhydrophobic vitrification process can eliminate cold chain entirely from the system. Basically, it is able to preserve proteins and nucleic acids at ambient temperatures using benign excipients and proprietary scaffolds. The technology is readily scalable for mass production and requires minimal reagent and capital equipment. So this makes it a financially attractive option compared to similar technologies right now, which are heavily revenue dependent. And then I'm going to pass it over to Dr. Pavanshu Mohanty to talk a little bit about the technology and the background of the science.
So the idea behind this technology is just turning the liquid that has the suspension of the molecule into a glass format. And the idea of a glass forming from liquid is not new. It has been used in many, many applications. And liquid nitrogen, slush liquid nitrogen freezing is one of the techniques that is used in biology and stabilizing biological samples. So essentially, the idea is you just reduce the molecular mobility, and you do it so fast that you prevent any crystallization, and it turns into a glass. And it is typically done by rapid cooling. But where our technology comes, it's actually the ambient processing technology, where we rapidly desiccate the carrier liquid so that we avoid or bypass any crystallization, and we form a gel or glass. So when I talk about the glass, I'm more talking about an amorphous material rather than the glass we typically know. And that is the background of the technology. And it is done by an excipient formulation plus a desiccation method that prevents any separation or crystallization. So can you go to the next slide? So the comparative technology, if you look at that, the lyophilization has been used. It's a gold standard in stabilizing drugs. So the primary process is basically you freeze, and then after that you do the primary drying and then secondary drying, and you end up in producing a powder product. In our case, what we do, we don't have any freezing involved. The process occurs at ambient temperature above the freezing point. And the idea here, essentially, you take the liquid formulation and rapidly desiccate them to form a gel or a glass. And there are significant differences in terms of, because freezing is not involved, there are many sensitive molecules that are actually get damaged due to freezing. So we can deal with many, many different biologics that are freeze sensitive. Then the processing time in lyophilization, optimization and control is a key parameter. And the key step, basically, that's where a lot of effort goes. In our case, The processing is very fast. Actually, depending on the type of biologic material, the process time varies from almost like 15 minutes to an hour max. And we can scale the process to very, very large amount of processing because freezing is not involved in that. So we'll show you some examples, what we have been working on. Laura, can you take from there? Yeah.
So the applications for this technology are very, very broad. They include a lot of your protein-based therapeutics, so antibodies, insulin. They also include assays, tissue biopsy preservation, vaccines, and blood products. So we're going to describe a little bit of an example using messenger RNA, especially since that is particularly important in terms of COVID-related vaccines. Messenger RNA are a prime candidate for multiple COVID-related vaccines, and they are also the ones that require storage at -70. So this is just an outline of our experimental process. So we have green fluorescent protein encoding messenger RNA. So essentially what this enables you to do is if you were to transfect cells with this messenger RNA, they would translate that into green fluorescent protein, and then you can image for the fluorescence using a fluorescent plate reader or using a microscope. So what we've done is we've taken this green fluorescent protein, messenger RNA that encodes the protein, we vitrify it using our technology. Then we stored the vitrified product or unvitrified control product at -20 Celsius, 28 and 55 degrees Celsius. Following storage, we then elute the messenger RNA that had been vitrified. We perform a lipofectamine protocol, and then we transfect CHO cells and detect the green fluorescence protein. So this bar graph is just data that's demonstrating our ability to retain green fluorescent protein messenger RNA expression following storage at various temperatures. So the y-axis indicates the relative fluorescent unit, so that's indicating how much GFP is there. And then the x-axis is broken up into two groups. We have our not vitrified group, so those were our control groups that were stored in the normal matrix that comes from the manufacturer at -20, at 28 or 55. And then we had the vitrified group where we vitrified the messenger RNA using our system and process, and then stored those samples at -20, 28, and 55. And what you can see is there's a red dashed line. So this data is presented as relative to as stored in accordance with manufacturer specifications, which is -70. So the red line is messenger RNA that was stored at -70. So that's our gold standard. And what you can see is even at -20, the unvitrified product, even after only 3 days of storage, had shown significant reduction in signal intensity. And that reduction only became more apparent with progressive storage. So 3 days, there was about 80% signal intensity. 7 days, that has dropped down to about 60%. And then by 14 days, that was 50%. And that reduction in signal intensity only becomes more extreme when you store at higher temperatures. So at 28 degrees, we're looking at about 50% signal intensity following 3 days of storage, and so on. Storage at 55 resulted in complete loss of messenger RNA expression and green fluorescence protein for all samples, even after 3 days of storage. Whereas for a vitrified product, you can see we retain green fluorescent protein expression for all storage temperatures from -20 all the way to 55, from 3 days all the way to 14 days. And we're currently doing a longer term stability study at this point. So that was just pure messenger RNA. So that messenger RNA wasn't stored encapsulated in lipofectamine. So a lot of the messenger RNA vaccines that are out there are encapsulated. So we wanted to do, this is a very early stage pilot experiment looking at can we vitrify and stabilize encapsulated messenger RNA. It's a very similar experimental design, except for this time, we performed the lipofectamine protocol before vitrification. So same product, GFP encoding messenger RNA, performed the lipofectamine protocol to encapsulate that messenger RNA, vitrified that product using our process, stored that vitrified product at -20 or 28, also stored unvitrified controls, and then we transfected CHO cells and detected green fluorescent protein by fluorescent plate reader. So this is our preliminary results from that work. And again, y-axis is relative fluorescent units. The two bars on the left indicate samples that were stored at 20 degrees. The two bars on the right indicate samples that were stored at 28. The dark blue is the unvitrified control. So this was a sample that was not vitrified and stored at -20 or 28. And then the orange indicates our vitrified product. So what you can see is messenger RNA that's been encapsulated with lipofectamine does have a reduction in signal when it has not been vitrified that our vitrified product is able to protect. And then 28, with storage at 28, you lose all messenger RNA encoding of green fluorescent protein. In our vitrified product, you retain that ability. So we're currently working on this further and exploring other lipid nanoparticles moving forward.
Thank you.
So that's our current state of affairs. We think that there's a lot of potential for this technology to reduce or eliminate cold chain requirements for a variety of products from vaccines, biologics, in a variety of temperatures, whether it's protecting from freezing or protecting from extreme environmental heat. And we look forward to exploring this technology further. And if you guys have any questions, please let me know. We'd be happy to address them.
Yeah, thank you so much, Dr. Laura, Dr. Pavanshu. I think huge potential, as you rightly said, waiting to be unleashed. I think some probably you might already be doing the chemical stability, the formulation, safety studies, and might want to get integrated with vaccines so that preclinical and clinical safety assessment get into way so that it can see light of the day for deployment quickly. If it works, you have it like $1 billion proposition sitting to be explored. Thank you so much. I think very similar technology I was told that Professor Raghavan is also working on. So maybe at this point I would request Professor Raghavan to come in and maybe share his expertise and his technology.
So, I will talk about work we have been doing on generating a thermotolerant formulation for a SARS-CoV-2 vaccine. And so everybody knows that there's been a lot of success recently with some phase 3 trial data, and there are a number of vaccine candidates in advanced stages of development. And virtually all current formulations require low temperature storage prior to administration. So there have been a number of formulations that have been shown to be immunogenic, both in animals as well as more recently in humans. And in animals, they have been shown to protect from severe lung pathology. And humans also, there's early evidence of protection. But how long the protection will last, we still don't know. And all the current formulations do indeed require low temperature, some lower than others. So we have been looking, like some others, at the receptor binding domain of the SARS-CoV-2 virus. So just a second. This is the spike protein of the virus. In red is the receptor binding domain, which binds to the ACE2 receptor. And in recent months, it's been increasingly clear that there are neutralizing antibodies which bind to many regions of the RBD. So colored here are different neutralizing antibody epitopes, and shown in blue are places where at least a month ago, neutralizing antibodies had not been found. So they're mainly at the N and C terminal regions of this. So what we did was to make a fragment of the receptor binding domain, and we put an extra glycosylation site at this region to shield it from eliciting what might be non-neutralizing antibodies. And we expressed it in mammalian cells, insect cells, and pichia, and found that the protein was highly thermotolerant as well as immunogenic. And in hamster challenge studies, which we have done so far with the insect cell expressed RBD, they conferred protection against weight loss and lung damage against very high titers of the virus. So this is just a little bit of the biophysical characterization of a couple of variants of the protein. They are monomeric, and they have a stability of about a TM of about 50 degrees or so. They bind well to the ACE2 receptor as well as another neutralizing antibody, so they are conformationally intact. This is the insect cell protein. We subjected it to various kinds of thermal stresses, and it retained its functionality. And once again, it shows the appropriate binding properties, and it's a clean peak by gel filtration. Next, we subjected the RBD to lyophilization and long-term thermal stress. So before and after lyophilization and redissolution, the protein remains intact, and even the lyophilized protein can be subjected to quite high temperatures for reasonable amounts of time. And in water, it remains stable at 37 degrees for a few days at least, which is about all we really need. But when lyophilized, we've subjected the protein to 37-degree temperatures for more than 4 weeks, and it retains its properties. So shown here are the binding properties, where again it shows very clean binding despite long time high temperature storage. So we also looked at the immunogenicity, and in guinea pigs, you can just look at the neutralization titers. So we got neutralization titers with a very stringent neutralization assay, which are comparable to several vaccine candidates, which are currently in various stages of clinical development. So the dashed line is where we are, and these are, so other than probably the Novavax, it's difficult to compare because these are all with different neutralization assays. At any rate, we next carried out a hamster viral challenge, and where we challenged with a high dose of the virus. And you can see that the G6 group, which is our candidate antigen, were completely protected. They gained weight. These are the days after challenge, like the unchallenged animals, whereas the challenged animals lost weight, and this is animals immunized with the full-length spike antigen. So in addition, we don't really have time to go through this, but the cytokine profiles were also favorable. And you can see here, these are the lungs from the immunized and challenged animals. These are from the control animals. So, and if you look at the viral load, there's essentially no virus in the lungs of the immunized animals, and these are in the control animals and with another antigen. So these animals were well protected from challenge. They showed no weight loss and very little lung pathology. And ones immunized with the spike ectodomain, surprisingly, didn't do so well. So this is again just a histopathology, and so in blue is with our antigen, and they showed much better scores with very little disease evidence compared to the virus control, which is in green. So I'll just conclude here. We designed a monomeric glycan engineered RBD fragment that is expressed in high yield in a number of different expression systems. It had identical thermal stability profiles before and after lyophilization. It's resistant to long-term thermal stress in solution, at least up to 50 degrees. And the lyophilized RBD was stable to transient exposure, even as high as 100 degrees centigrade. And at 37 degrees, over 4 weeks, it retained all its properties. We expressed similar protein in Pichia, but there the results were unfortunately not so good. And the sera in guinea pigs neutralized SARS-CoV-2 virus with quite high titers. This is against replicative virus, not pseudovirus as many other studies use. And in the hamster challenge studies, they showed very good protection. And essentially, such thermotolerant RBD derivatives show a lot of promise to combat COVID-19. So this was done with a number of collaborators from my lab, ICER Trivandrum, THSTI, and Minvax, which is a Bangalore-based startup, which was really instrumental in carrying out all the guinea pig and mouse immunizations and the serology studies. The challenge studies were done at THSTI. So this is all published in JBC, and it really has received a lot of attention in the media.
Indeed, Professor Raghavan, I think it was a wonderful work and a very good lead that we will all wish you good luck to develop and translate soon.
Okay.
At this point, I would request my next panelist, Dr. Suman, who is a professor at IIT Kharagpur, and he and his team is developing an improvised next level of LAMP-based diagnostic test. And we would love to hear the advantages over and above the existing diagnostic kits. Thank you for waiting, Dr. Suman. Over to you.
Yeah, so thank you so much. I just want to share my screen. Thank you very much. I am grateful to the organizers, and to be very frank, I'm not an expert in vaccine development, but I primarily work in diagnostic technologies. But I have listened to all the expert talks on vaccine development, and interestingly, I find a lot of synergistic similarities between various aspects of developing diagnostic technologies and vaccination technologies. And I presume that these technologies need to go hand in hand because vaccination is important, very, very critical, but diagnostics also should continue. So the major challenge that we have realized over the years in the Indian scenario is the extreme centralized facilities in specialized healthcare, which means that majority of the nation is deprived of even the very basic primary healthcare amenities and facilities. So these will include everything starting from basic vaccination to diagnostics to advanced treatment. We all talk about the shortages of doctors, nurses, technicians, other barriers to access, and several other socio-economic factors. But we often forget that technology happens to be the most critical barrier here, because the conventional ones do not work in these extreme point of care environment. So I just wanted to give this little bit broader perspective, because the issues of vaccine delivery and issues of diagnostics can be connected by this broad philosophy or structure of a rural health kiosk in which I personally work as a team member in coordination with medical doctors and other entrepreneurs. So this is essentially an electronically enabled support system via telemedicine, where the primary health workers interface with the patients, tabulate or enlist the issues faced by the patient in the form of a user interface in a tab or a laptop computer, or it could be a smartphone, and these are disseminated to experts who are sitting in the big city, and the advisory of the expert at the first instance is commonly some drugs, could be injection, could be oral, and of course some diagnostics. Now there comes the bottleneck, because there are issues associated with inaccuracy, delay to distance related factors, and issues of reagent stability. Why I am talking about these is because some of these issues are equally important for diagnostics and also important for vaccine delivery. The innovations which are specifically required here include the non-availability of high quality power, the environment where we have extreme heat, dust, humidity. The reagent storage, which is so critical for vaccination, it is equally critical also for diagnostic master mixes or kits. So the specialized air conditioners and refrigerators for controlled environment, they are not there at resource limited settings, and this could lead to degradation of reagents. The supply chain, which is very important again, both for these kind of disruptive diagnostic technologies as well as vaccination technologies, it may not exist. Trained technicians may not be available, and price sensitivity is very critical. So keeping all this in mind, when we started looking into the COVID-19 testing, I mean, before that I had a significant experience in developing diagnostic devices for rural healthcare, but primarily for non-viral testing. But this was the first time I was doing a viral testing, and I realized that an obvious easy solution could be the rapid antigen based kit, the so-called rapid kit, which gives very quick results, but the results are not very reliable, typically at early stages of the detection. Therefore, we decided to go for a technology for detecting the viral RNA, but somewhat different from the current diagnostic tests which are available. In this perspective, because there are so many tests, so many technologies, so far as I understand, currently more than 600 numbers of COVID tests, different kinds of COVID tests are already there in the market, but people are confused whether the test is screening or confirmatory, whether the cost of the kit is representative of the cost of the test, or there are other hidden costs. Cheaper the better, quicker the better. Are there compelling issues of reagent contamination? Are the reagents stable? Are there cross reactivities? Are the methods resource intensive? Are they user friendly? In the act of making them user friendly, does it still remain sensitive and specific? And are we unnecessarily using fancy electronics or indirect sensing and deviating too much from clinical gold standards? So in the way, what we wanted to do is summarized in the form of this slide. We all know that the RT lamp based technologies, the so-called isothermal technologies for rapid diagnostics, these technologies are primarily based on certain steps which are conceptually very similar to what is done in the RT-PCR test. That is, you still extract the RNA, either you extract or you isolate by certain simplifications from swab or saliva. You still do the amplification, that is, amplify. Instead of using a thermocycler, you use isothermal technology, and still you have to detect the amplification products. We wanted to combine all these with a very specialized DNA based probing step, which is very different from the RNA based probing step that the CRISPR or the so-called Feluda kit and the other devices do. So this is different from CRISPR in the way that it is a DNA based specific probing which enhances the specificity of the test, at the same time doesn't compromise with the stability, which is which may be vulnerable if it is RNA mediated, and the entire collection of events starting from sample to the solution is integrated in this portable device which we call as BIRAC box. So here we have a pre-programmable unit where you could pre-program the electronics of this unit and the thermal unit so that you could have as many number of isothermal steps in the reaction, in which there will be reaction participating in this specified isothermal condition, followed by a seamless dispensing in a paper microfluidic strip for smartphone based readout and dissemination. So as per ICMR guidelines, this was tested and validated at the ICMR NICET, certified ICMR lab to do this validation. And the results were very, very encouraging, and we got a very high level of sensitivity and specificity, considering the extreme simple nature of this device, which in US dollar costs within $100 cost per instrument, which is a fixed cost, and then per test it will be around $6 to 7 US dollars. So we wanted to have a journey of this from lab to the field. Because of interest of time, I will not show the video, but the entire crux of the story is we wanted to bring down the extremely advanced premises of molecular diagnostics from high-end lab to the field where there are extremely limited resources and uncontrolled laboratory environment. So in this way, not only for COVID-19, we wanted to have a paradigm shift with a simplified device unit for a RNA-based diagnostic procedure. Today it is COVID. Everybody is bothered about COVID. There are other diseases which might be equally important and severe for some individuals, and tomorrow it will be something else. So once we have such a structured machine unit and a generic method, the technology that comes in the backbone is not just going to help for us to face these kinds of pandemics in the long run, but in the process, these technologies may also be effective in simultaneously developing, the storage, the specialized reagent management and supply chain, also for developing novel vaccination strategies. So I wanted to acknowledge my entire team who did this work, and of course the ministry, the government of India, and the ICMR unit who did these validation studies. So thank you very much, and it was a pleasure in presenting my work.
Thank you, Dr. Suman. Thank you for taking us over a platform technology that can be made more affordable and simplified, covering the last mile. We wish you good luck to get it rolled out. And in the audience, anybody interested in technology transfer for this, Dr. Suman would be very happy to interact with. I think I thank my panel speakers. We were able to bring a glimpse of the innovation quotient which is thriving in the to be unleashed and to be recognized, and all the examples starting from the LAMP PCR test that we just talked about, promising lead candidate Professor Raghavan spoke about, the Akara's excipient which can obviate the need of lyophilization or keeping cold chain deliveries at minus 50 degrees to ambient temperatures. And in the existing solutions, existing vaccines rightfully delivered at cold temperatures through Emvolio and Selfshare technologies. And at the mass vaccination scale, we can also recognize the relevance of safely injecting using safety syringes that can address the limitation of use of overages in the unit doses, and we can look at bulk and cartridges. cartridge deployment at multiple dose level. So I think there is a great potential, and I firmly believe that these expectations can be met and the new opportunities can be created from many such affordable, useful solutions which can emerge from Indian innovation ecosystem. So congratulations to all the organizers for putting such an valuable theme for the conference, and I think it was a very knowledgeable, and I personally learned a lot through this conference. Thank you.